Our next speaker is going to look at some technologies to capture some of these biological differences. Eric Schadt from Pacific Biosciences and Mount Sinai School of Medicine. Eric?
All right, thank you. So we've been focusing a bit on the types of technologies that today are pretty fascinating because they are enabling us to peer into single molecules, single biomolecular machines in real time and observe what they're doing at the very fundamental level, whether it's making DNA, RNA, proteins, and then how these all fit together to give rise to higher complexity in life at the cellular and higher order levels. And these sorts of things can now, as you've heard talked about, be scored in very comprehensive fashion in individuals. So whether we're looking at DNA sequencing or RNA sequencing or metabolite or proteomic profiling, all of these different dimensions of data can be scored in a very comprehensive fashion. And now with the technologies for low cost, we can do this over many individuals in a population, not just single individuals. And the aim, of course, is to integrate all of this information to define the complexity of life. And the way I like to depict that is in this animation, where we're not only looking at things like DNA in one dimension in isolation as a linear system, but we're looking at DNA and metabolites and proteins and so on in the way in which they interact in highly nonlinear ways in these living systems. And they form these networks of highly interconnected sets of genes and proteins and metabolites. And these networks aren't acting in isolation either, right? They're acting as a concert of networks. And this is what is going on in the system that gives rise to all the higher level complexity. And once we understand these sorts of networks, we can start looking at the subnetwork, get away from this one-dimensional single gene view and start looking at how changes in one part of the system, in one functional unit, one subnetwork cause changes in other parts of the system. And this may be going on within a given cell or a tissue type, and we're more complicated than a single cell or tissue type. So we want to start modeling how are these perturbations that are either genetically or environmentally induced being transmitted across the system between different cell types, between different organ systems to coordinate a system to live the way we all want to live. So once we have this sort of model, once we have this modeling, the right data generated, now we can go into a more in silico prediction-based biology where we can use these models to understand how perturbations in one part of the system, again, whether it's genetic or environmentally induced, get propagated throughout the system and what kind of steady state does that system achieve at the end of the day, and is it pro or against disease. So the technologies, one of the dimensions I focus a lot on, the DNA RNA dimension, has over with PacBio has focused on a new kind of sequencing instrument that's peering down to the DNA polymerase and looking at a single molecule of DNA polymerase as it's synthesizing a strand of DNA. And that's, of course, leveraging what nature gave us as a sequencing instrument, given that's in all of our cells replicating our DNA on a daily basis. And how do we achieve looking at a single molecule of something in real time to observe what it's doing? And the way to do it is to isolate these single molecules in what we call a zero-mode waveguide. So it's a very tiny nanometer-size hole that's been etched into an aluminum film that's fixed to a solid glass substrate. And it's about 100 nanometers in diameter, about 100 nanometers tall. And by confining down to this very small space, when you fire a laser on the underside of this, because the visible wavelength of light is much larger than 100 nanometers, no standing waves of light can be guided through that waveguide. So they instead decay exponentially into the hole and give you a 10 to the minus 21 liter confinement volume, which is small enough to basically ignore all the other stuff that's going on in the system and just peer down at that single molecule and directly observe it. And that's a single zero-mode waveguide, and what we can do is achieve multiplexing by looking at multiple zero-mode waveguides at a time. So what I just showed you was a single zero-mode waveguide, and what we can do is look at tens of thousands or hundreds of thousands of these reactions going on in real time in the system to achieve a higher throughput scale for determining the variation of DNA in the system. And I'll just give you a few highlights of why this kind of how is this technology changing? How is it different than next-gen sequencing technologies, and why is it changing what we can understand about biology? So I'll highlight a paper we just had come out in collaboration with Neil Schadt at UCSF in Nature. It was out, I think, yesterday. So this was on looking at FLT3 and the association to AML with variations in FLT3. And AML, of course, not a good disease that you want to have. And FLT3, activating mutations in FLT3 were known to associate with AML. And but all the it's a tyrosine kinase, and of course the kinase inhibitor trials all basically failed to achieve any efficacy through their development. And so the question, the dogma that started to emerge in the field was, oh, this is this activating mutation in FLT3 is really just a passenger lesion. It's not really causally involved in the AML. So what Neil's lab did at UCSF is they did a mutagenesis screen in a cell line and treated with different tyrosine kinase inhibitors to basically lock onto variations that were being induced that were leading to resistance of that drug. And so here's just some of the data for that, some of the codon changing mutations that they uncovered in this mutagenesis screen that were definitely affecting the effects of the drug in the cell survival. And so clearly mutations were capable of leading to resistance of that particular treatment. And so what was interesting is the activating mutation, which is basically a 20 to 100 base pair repeat, occurs in this part of the gene. And the different mutations they were uncovering that led to resistance in the cell system were falling downstream of that, some of them up to 800 base pairs away from that activating mutation. So one of the benefits of leveraging DNA polymerase as your sequencing engine is you get the processivity of the enzyme. You're not constrained to these short reads that are anywhere from 25 to 500 bases long. We're seeing things that are 3,000 to 20,000 bases long. And so the idea was we could bring long-range sequencing to bear in this sort of problem and not only completely resolve the structural variations that occur in this gene that associate with AML, but to directly phase those on a single molecule level, right? The fact that we could see the activating mutation and on the very same molecule see the DNA mutation, that enables to directly phase that information in that context where we're able to resolve the efficacy problem. So just to again highlight the advantage of looking at these longer reads with short read technologies like Illumina and even medium range technologies like 454, you're not able to, with the shortest reads, detect the ITDs in these activating mutations in an error-free way. You're certainly not able to phase those distant mutations. The same holds for 454, and you're not able to, in the case of Sanger, if you want to use Sanger sequencing for their longer reads, able to detect the low frequency mutations. And so the home run was we sequenced this gene in populations that had been blood collected before they were put on AML patients, before they were put on treatment, post-treatment, set up the sequencing system to basically read through the entire gene, right, in a single reads, capture all that information. And the results were pretty stunning in that the resistance mutations that Neil's lab uncovered through the mutagenesis screening were basically non-existent in the patients before treatment versus post-treatment, some of those popping up at greater than 40% frequency. So a very clear indication that FLT3 is absolutely a target for AML. And what's actually going on is these forms of the gene that have these certain mutations in them defeat that treatment, and they proliferate and become the dominant forms in the system, and you develop complete resistance. So sort of overturning that emerging dogma, again, just through a technology that enables you to see the real picture, see the real biology going on in the system. So I wanted to also talk about our understanding of DNA from the standpoint of what constitutes DNA. And we all like to think about the A, G, C's, and T's. But what I'll show you next is how these technologies are uncovering whole new dimensions of the chemical structure space within the DNA that I think is going to have a profound implication on how we really assess variation among individuals in the population. And I'll start with what we had done with looking at DNA from bugs in the environment, obviously something that's important to us all given we're being bombarded every day, and these are things that make us sick every year, from cold and flu to more severe illnesses. And we had a paper in New England Journal late last year on this E. coli outbreak in Germany, where again we leveraged this new technology to very rapidly in a single day sequence not only the outbreak strains that were the outbreak strain from isolates from people who were infected in Germany, but all the other similar serotypes, bacteria with a similar, E. coli with a similar serotypes that didn't achieve the same virulence that the outbreak in Germany did. So the outbreak strain in Germany was particularly virulent, right? It caused this severe kidney damage. There are roughly, I don't know, something on the order of 1,000 individuals in Germany today on dialysis because they were infected with this bug. So the question was, how did it achieve such virulence when all the other bugs with the same serotype haven't been observed to be so virulent? So we, in a single day, sequenced on the order of 9 strains from the outbreak and then other serotypes collected from around the globe, completely sequenced those genomes, completely de novo assembled those genomes, and then did this comparative analysis, not only with strains of the same serotype, but all the other bugs of E. coli that have been sequenced, and clearly placed the outbreak strain in this group of serotypes. If you remember, there was a controversy, BGI, Beijing Genome Institute, and a group in the UK published a paper indicating that this particular outbreak strain was actually an enterohemorrhagic E. coli strain, not a enteroaggregative E. coli strain, and actually published papers on that, and our data show that's absolutely incorrect. By, again, generating the complete assemblies of these genomes, we were able to show that it's absolutely an enteroaggregative E. coli strain, not as closely related to these green guys, which were the enterohemorrhagic. And based on that, we were able to start doing this comparative analysis where we were looking for, so these different concentric circles are the different strains that were sequenced in the O104 serotype group. And so you're just looking at the different genomes. And now what we're doing is a comparative analysis where this is a Circos plot where we're color coding by coverage to a given reference. And the main thing to point out is this red indicates regions of the genome that are absent with respect to the reference, and blue-green means you're very similar to the reference. And what we see is this outbreak strain, which is a second concentric circle, has this particular piece that's present in it, but absent in all the other strains. So that was turned out to be a prophage element that contained the Schadt toxin gene, and of course, Schadt toxin known to increase virulence of these bugs. So that was sort of one of the smoking guns we uncovered. So we not only uncovered sort of how the virulence and how it came to be, that this was an enteroaggregative strain that acquired lots of different elements from enterohemorrhagic strains like the virulence plasma, antibiotic resistant plasmids, and so on, through horizontal gene transfer, acquired these elements, became very virulent. We also predicted that the effects of treatment with antibiotics, certain antibiotics like Cipro, would not improve the condition at all. It would actually worsen the condition, and we published this in our paper as well, that this is the Schadt toxin expression in the presence of Cipro versus control, an 80-fold induction in the expression of the Schadt toxin gene in the presence of Cipro. So that's not only was it resistant to that antibiotic, but it activated the Schadt toxin gene. So not a good treatment to put people on. So that was sort of what we published in New England Journal, but we have a new paper coming that'll show sort of completely flip everything over on its head, because what we can do with the data that was already published is, PacBio, because we're looking at the enzyme as it's sequencing in real time, we're picking up all this kinetic information. And the kinetic information, this enzyme is exquisitely sensitive to the sequence context of the sequence it's synthesizing. So variations in that context cause changes in the kinetics of the enzyme. And so how can we use that to uncover a brand new world of chemical modifications in these bases? Well, here's an example of how it works. So here's a color trace coming off the machine, sequencing a strand of DNA. This is time on the x-axis. This is intensity of color on the y-axis. And these different pulses, or different color bursts of light, are indicating what different tag nucleotide is being incorporated in real time as the enzyme is sequencing the DNA. But what you notice from this is all the kinetic information. You're seeing how long did it take that enzyme to incorporate that residue? How long did it take to translocate to the next base? And now just look at the comparison of what happens. So these are two identical strands of DNA with the exception of this A residue being methylated. And look at what happens when that enzyme encounters a methylated A residue versus a normal A residue. The time it takes to translocate to the next base is much longer. So this shift in kinetics can serve as a surrogate for chemical modification of these bases. In each of the different chemical modifications that are known to occur, every single one of these, whether it's 5-methyl-C, which is the most common, right? The only thing that existing technologies today can pick out on a genome-wide scale is 5-methyl-C, right? Through things like bisulfite sequencing or pulling regions down that are so modified. But things like hydroxymethyl-C, 4-methyl-C, 6-methyl-A, J bases, 8-oxo-G, ribonucleosides, all of those different things that are known to occur are undetectable with current technologies. But with the PacBio instrument, given the shifting in kinetics, we can see every single one of these chemical modification types. So what can we do with that kind of information? So the first thing I did was I said, okay, let's go back to the E. coli data and let's run the little model paper I have coming showing how we actually detect these modification events. So applied this model to the E. coli data, and what you're looking at here is the E. coli outbreak strain chromosome, and the red curve is minus log 10 of the p-value of the statistical model I applied to detect these kinetic variation events. And this thin black curve is a 5% false discovery rate line. So every time that minus log 10 of the p-value bounces above that line, that's a genome-wide significant event. And the first thing you notice is, wow, look at all that chemical modification going on. It was almost shocking, the scale of the signal. So it was so big we had to put our little thinking hats on to say, how are we going to resolve what all of these are? These real chemical modifications? What's going on? So the first thing we did was focus on the A residues. Why? Because we know that this particular strain of E. coli contains DAM methylase. This is an enzyme that's known to modify the A residue in the GATC context. So the first thing we did is looked at, well, how many A residues of all the 100,000 residues that we found modified were A residues? 50,000 were A residues, so very significantly enriched. In 93+% of the occurrences of the GATC context in that outbreak strain were detected as being chemically modified. So a great positive control. That's sort of what you'd expect with DAM methylase in the system. But it still left 12,000 other methylase or A residues that weren't explained by the DAM methylase. So we started looking for patterns of other sequence context in that genome. And what we found was a whole world of different motifs, some of which had never been seen before as being chemically modified, popping up. For example, 94% of the occurrences of the 6-mer motif were detected with the A residue being modified. Right? The p-value, that kind of enrichment occurs by chance, is less than 10 to the minus 300th. Right? It's a very statistically significant event, even after corrections for multiple testing. So 93% roughly of the ACC context, this A residue was detected as chemically modified. Again, very, very significant. This motif never before seen in nature as being chemically modified, but we were making the detection loudly and clearly. So then we went to my friend, Rich Roberts, who is a Nobel Prize winner, is now at New England Biosystems, focuses on methylases. So said, could you run your bioinformatic analysis on the genome of this bug and tell us whether there are other methylase-like genes that might help us explain the patterns we were seeing. So he ran his analysis, found 11 methylases, which he thought was quite surprising. So genes that look like methylases using his bioinformatic analysis. Then what he did was he cloned out every single one of those 11 guys into a plasmid, banged that plasmid into a methylase-free strain of E. coli. So the only methylase activity going on would be the methylase he banged in. And then we simply sequenced and saw what kind of signal are we seeing for the chemical modifications. And what we saw was awesome. It was we directly mapped all of the motifs we were detecting in a completely data-driven fashion to the genes that were making the modifications. So for example, he predicted this methyltransferase at this particular position in the E. coli genome, banged that into the methylase-free strain. We did the sequencing of the plasmid. Each one of these spikes corresponds to a very strong modification signal, and they all reflect the ACC modification, this completely novel modification. Now we know the gene that's making it. The same thing for the CAG. We found the enzyme that was making that particular modification. Darn, I left the slide out. I have another, one of the other methylases was modifying every single A residue in the genome, right? Like, so that sort of non-specificity hadn't been seen before, yet it was turned off in the gene present in that outbreak strain, but not being transcribed. So interesting to think about the switch that E. coli throws when it wants to modify all of those. The most fascinating thing to us, though, was the CTG-CAG motif. The enzyme we found that was making that modification fell right next to the Schadt toxin gene that was dropped in by that virus, right? That bacterial virus dropped in not only the Schadt toxin gene, it also dropped in this methylase that was making modifications over the genome. So then we RNA sequenced these guys. We knocked the methylase out, did the RNA sequencing compared to the wild type, and to see functionally whether we could see anything going on. And surprisingly, there was a pretty smashing signature that was very, very enriched for genes that were upregulated. So again, this is going against all the dogma in the field about what these modifications are doing. So here what we're seeing is if you knock that methylase out, you lose that modification, you've upregulated a whole host of genes that are enriched for many different pathways, some of which are involved in horizontal gene transfer, the very pathway we hypothesized was involved in the increased virulence of the strain. So now we have pretty hardcore evidence that there's that particular perturbation is causing changes throughout the genome, and then we want to actually do the validation of throwing that into an animal model and seeing whether if we knock out that methyltransferase, it has an impact on a model. So Matt Waldor, who's been my partner in crime on this whole thing, has an evolving rabbit model for E. coli infection. The animals get, inflammation in the intestine and diarrhea just as we do. And so he infected the little rabbits with the wild type strain and with the knockout version. And 100% of the animals infected with the wild type strain got sick. 100% of the animals infected with the knockout methylase did not get sick. So pretty intriguing. That's still undergoing validation. Like, I won't claim that's the end of the story, but pretty amazing to see that sort of effect on higher order phenotype. And so just to sort of summarize, my little movie will play. It's thinking about it. So just to summarize the sort of the whole train of what's going on here is we have, right, bacterial virus punching into the bacteria, dropping in its genome, and that genome integrating into the host genome. As a result of that integration in the genes that dropped in, it then causes epigenetic changes throughout the host chromosome. As a response to that epigenetic change in the host chromosome, it perturbs a number of different pathways and networks that affect higher level functioning, and perturbations in those pathways lead to increased virulence and other effects. So, that's the deal. One of the things I've been thinking about for how to apply this more generally, so the things with the E. coli and the cholera outbreaks we published in New England Journal on was that people were already showing up to the hospital sick. So we've been thinking about real-time pathogen surveillance in areas like airports here in San Francisco, or your different food stores and restaurants. Can we assay these environments in real time, sequence that information, and understand the flow of pathogens through geographic regions over time, sort of look at pathogen flux over time. So that's sort of active stuff we have going on. And we have carried out a number of pilots that have shown our ability to do this in real time here, focusing on a metaviral genome pilot where we looked at all the acute viruses of public health concern. We were wanting to sequence them in areas we assayed around the Bay Area. One of the first things we did was focus on sewage, and here's the Bay Area where this red dashed line splits the city in two. If you live on this side, your sewage goes over here. If you live on this side, your sewage goes over here. And people were always asking, why the hell are you sequencing sewage? And the answer came very simply from my youngest two daughters and one of their favorite books called Everyone Poops. And it's because if you want to cast a net over a large geographic region, sewage is an excellent thing to look at because everybody contributes to it, and they contribute every single day. So that was the motivation. We also were swabbing surfaces money, telephones, desktops, doorknobs, you name it. We were swabbing it over time. Did nasal swabs as well in volunteers from PacBio. And the main take-home of all this, since I'm basically out of time, is that we were able to resolve, the sewage was pretty interesting because not only were we picking up respiratory viruses and other things that, were questionable whether they'd show up in sewage, but we were picking up viruses that were specific to the different foods you eat. So things like peppers and tomatoes and chicken and other food types were showing up as well. So started thinking in the molecular epidemiology context, could you start assessing the diet composition, say, of a region by sampling at higher density substations and then correlating that with global health of the region? Just things that we haven't thought about before. Found lots of different things on the surfaces. We're able to loosely correlate increasing load of influenza virus on surfaces during the course of an actual outbreak of flu in that environment. So again, using bugs as sensors in the environment, I'll just close with this little scenario of how I see this all potentially helping public health and environmental surveillance. So imagine your little neighborhood here in Palo Alto, sunny Palo Alto, in your house over here, and in your house you have your fancy toilet. I'm a big fan of the Japanese toilets in Tokyo. If you ever go there, all the buttons they have when you can't sleep, it's a fun little thing to do. And some of those toilets will collect samples for medically relevant testing. Right? That's pretty interesting. So you can imagine collecting samples now from these households and popping it into the sequencer and sequencing what's coming out of that house and to understand, again, not just things that are causing illness, but using these bugs as sensors, right? If you take a look at the E. coli thing, the different chemical modifications that were occurring, they may be sensitive to different toxins in the environment and so on. So thinking about making those sorts of maps, and what the heck are you going to use that for? Well, I like to paint this little scenario of your son calling from Palo Alto High School, saying he wants to go over to his friend's house to play, and the mom's, let me pop this up on the computer. And so she goes and pulls up Billy's house in Google Maps, pulls up the metagenomic report, and what she sees is there's no acute infections going on in the house, but she's like, my God, look at that microbiome composition. It's pro-diabetic, pro-atherosclerotic. So she says basically you can go over to his house to play, but you need to come home for dinner. So those are and the main take home there isn't to cause fear about all of this, but to show you that this sort of information is directly actionable. It's actionable today. If we can, generate this sort of information on a routine basis, the cost points are getting to a place where it's a reasonable thing to do. And if you knew a pathogen was flexing into your house, a certain strain of E. coli that's known to be very virulent, or a influenza, maybe you go out and buy Tamiflu, maybe you throw out your food. If you see cancer cells, colon cancer cells being excreted, those are the sorts of things you're going to take action on. So it's something worth thinking about. So I wanted to just thank the crew at PacBio is awesome. We've had a lot of fun there. Lots of different academic partners making all this work possible. And we're now, of course, all setting up at Mount Sinai to continue our fun. So thank you.
So George Daston, Procter & Gamble. So the idea of sequencing sewage, suggests, one of the things that are always evolving along with the bacteria are bacteriophage, and those might actually be easier to sequence and might actually give you a quicker read as to evolution of novel strains. Did you find phage when you were doing the sewage treatment, and did you do long enough time course to see that sort of coevolution?
Yeah, I totally agree with you. In fact, we started this was with Joe DeRisi at UCSF, who really had his guys out there in the sewage plants collecting samples. And the aim was to focus to viral particle sizes, so there was a big filtration steps to get down to really small sizes. But if you look at the taxonomy, we were picking out lots of prokaryotic and eukaryotic DNA, so lots of it was going through, and a lot of it was prophage, and so that did turn out, and highlighting that here, that did turn out to be pretty interesting. We didn't sort of have the density of sampling to establish baselines and look at fluctuation from the baseline to see how it associated. But that's all work we have ongoing as a next step. Totally agree with you that that's a good angle to pursue.
Hi, Nat Rothman from NCI. I was wondering what your throughput capacity is for this technology, specifically in the context of the capability to do studies in humans where there might be 200, 400, 600 samples of, let's say, buccal cell samples from mouthwashes and prospective cohort studies. Can you handle, I mean, the technology is really striking, can you handle hundreds of samples in a timely and not too expensive manner?
Right. So the big downside, like, why the hell am I focused on bacteria? My whole history had been in human diseases, and the bacteria was really a function of throughput of the system. So the downside at this point with PacBio is the throughput. The number of holes you can read is a function of is what defines the throughput. And right now, the camera sensing technologies are such that you can only get roughly 100,000 holes, so you don't quite get the throughput that you get on the current second-gen technology. So that is a big limitation for this type. This stuff was all targeted. The viral stuff. That's again why we started viral, because we could directly target these panels and then sequence what came out of that. So you're getting about 250 megabases or so off a chip today. That costs you $100. To generate that data. So you can sort of run those numbers and figure out, at this point, for human or mammalian system studies, it's really targeted. You have to be targeted. One of the things that might interest this crowd that I'm doing as a follow-on to this epigenetic stuff in the E. coli is mitochondria. And we are seeing amazing epigenetic changes going on in this mitochondria. Again, stuff you've never been able to see before because the technologies didn't exist. But seeing greater than 3% of the mitochondrial genome epigenetically modified and affecting all sorts of genes that are known to associate with hundreds of different disorders. So I think targeted panels is really the.
So if you targeted only to the mitochondrial genome in human samples, but comprehensively assessing it, since it's a lot smaller.
Exactly.
Targeted not the particular parts of the genome, but just the mitochondrial genome.
Just the mitochondria.
How many samples could you do a day?
So to do a mitochondria, we can put roughly 5 to 10 of those on a chip. That chip, again, costs $100. Takes 45 minutes for the run. So, again, you can do the math.
Yeah.
You can do that. So, again, it has that advantage that it's really granular. So, you can put those sorts of, like sequencing panels of genes, like 10 to 100 genes, you could do in that way as well from human. But whole human genomes is we're a couple years away from that.
Thank you.
So thanks to our speakers, I think we had 3 very good talks to show the diversity of this particular issue that we're faced with and the technologies that are available to us. We're going to take about a 10-minute break now. Get up and stretch and get a cup of coffee, and we'll come back in about 10 minutes, say 10 minutes to 11.